We are back with another series. Let's revise PYQs. This will benefit you in your upcoming exam. Let's look at the first question from this video. Question 1. Which of the following substances inhibits cytochrome complex 4, a component of the electron transport chain responsible for ATP synthesis? Your options are, and the correct answer is cyanide. Here's the explanation.
Electron transport between complex 4 and oxygen. Between complex 4 and oxygen is inhibited by hydrogen sulfide, cyanide and carbon monoxide. All common poisons including hydrogen sulfide, cyanide and carbon monoxide. So if they ask you cyanide causes what kind of toxicity, what should be your answer? Cyanide inhibits electron transport between complex 4 and oxygen. So tissues will not be able to use oxygen and that is called as histotoxic hypoxia. What does cyanide cause? Cyanide causes histotoxic hypoxia wherein however higher oxygen is available, your tissues will not be able to extract oxygen because electron transport chain is inhibited by cyanide. So cyanide causes histotoxic hypoxia.
Question 2. Which of the options aids in the transportation of fatty acids through the inner mitochondrial membrane? Your options are, and the correct answer is carnitine. Here's the explanation.
Now, with this background, let's try to understand what is the fate of any fatty acid that gets into any cell. For now, forget about fatty acid oxidation. We have just taken a fatty acid into a cell. What do you think will be its fate? As soon as fatty acid gets into any cell, irrespective of the final fate of fatty acid, the first enzyme to act on this fatty acid will be acyl-CoA synthetase. And this acyl-CoA synthetase uses one ATP which gets converted to AMP and PPI. So just this step uses two high energy phosphates and we successfully convert fatty acid into acyl-CoA. Didn't I tell you something similar for glucose? I told you as soon as glucose enters into any cell, irrespective of the final fate of glucose, the first enzyme to act on glucose would be hexokinase or glucokinase, converting glucose into glucose 6-phosphate. And I told you the purpose is to trap glucose inside the cell. So what do you think is the function of acyl-CoA synthetase as well? The purpose is to trap fatty acid within the cell. Because as we all know, fatty acids are non-polar. It's a lipid. So fatty acid is a non-polar substance. So it can easily cross a cell membrane, get into the cytoplasm. After your fatty acid is entered into cytoplasm, if you don't convert fatty acid into any other form, the fatty acid concentration inside the cell becomes higher and starts getting effluxed across the membrane back into the circulation. Can you afford to miss fatty acid this way? No, you have taken fatty acid for a reason. And that is why to trap fatty acid within the cell, you don't mind wasting two high energy phosphates and you convert fatty acid into acyl-CoA, which is polar. Once it becomes a polar form, it cannot go past the lipid bilayer. Got it? So the purpose of this step is to trap fatty acid within the cell. And after you form acyl-CoA, further fate of acyl-CoA will depend upon the energy status of the cell. Just think and tell me what is expected to happen to your fatty acid if the energy status of the cell is already high. If the energy status of the cell is already high, will you want to waste the fatty acid by oxidizing it? What is the purpose of fatty acid oxidation? It is to provide you with energy. But if the energy status of the cell is already high, will you want to waste the fatty acid by oxidizing it? No. What will you try to do with the fatty acid? You will try to store the fatty acid in the form of triacylglycerol or as cholesterol ester so that you can use it in future if there is a need. The energy status is already high. You will store the fatty acid as either triacylglycerol or as cholesterol ester. But what if the energy status of the cell is low? In that case, you want your fatty acids to be oxidized. And for your fatty acids to be oxidized, your fatty acid should cross the inner mitochondrial membrane. As acyl-CoA, which is polar, it cannot cross the inner mitochondrial membrane, so you need a carrier. And which carrier carries fatty acid across the inner mitochondrial membrane? I told you in the session on fatty acid synthesis as well. The carrier is carnitine. Okay. So what is the enzyme now? In the next step, carnitine reacts with acyl-CoA. It gets converted to acylcarnitine. And the enzyme is carnitine acyltransferase 1. And then this acylcarnitine crosses the inner mitochondrial membrane and it gets into the matrix. And once this acylcarnitine has entered into the matrix, if you retain the carrier, the same carrier will take the acylcarnitine back into cytoplasm. Can you afford to miss this fatty acid back into cytoplasm? No, you have taken fatty acid into mitochondria so that you can oxidize it and get the necessary energy. So you don't want the carrier, so you're going to release the carrier back. And that is done by carnitine acyltransferase 2. So what does carnitine acyltransferase 2 do? It releases the carnitine back. Yeah, it releases the carnitine back and it converts acylcarnitine to acyl-CoA. Now that you've formed acyl-CoA within the mitochondria, it cannot go past the lipid bilayer because I told you acyl-CoA is polar. And it gets trapped within the mitochondria where it gets oxidized. Got it? So this is the fate of any fatty acid that enters into any cell. I'm going to repeat it quickly. As soon as fatty acid enters into any cell, irrespective of the final fate of fatty acid, you have to trap the fatty acid as acyl-CoA, and the enzyme is acyl-CoA synthetase, which uses 2 high energy phosphates. After you formed acyl-CoA, fate will depend upon the energy status of the cell. If the energy status of the cell is already high, no point in oxidizing it. You have to store it. So you'll store fatty acid as triacylglycerol and as cholesterol ester. What if the energy status of the cell is low? Then the fatty acid has to be oxidized, for which your fatty acid should cross the inner mitochondrial membrane. As acyl-CoA, it cannot cross the inner mitochondrial membrane. You need a carrier. What is that carrier? It is carnitine. And what is that enzyme? It is carnitine acyltransferase 1, which converts fatty acid and carnitine to form acylcarnitine. And until carnitine acyltransferase 1 acts on fatty acid, have you committed fatty acid to any of the pathways? No. The fatty acid can enter into cholesterol ester formation, or it can enter into triacylglycerol synthesis, or it can get oxidized. But once carnitine acyltransferase 1 acts on acyl-CoA, converts it into acylcarnitine, no other go, it goes and gets oxidized. So which enzyme commits fatty acid to oxidation? It is carnitine acyltransferase 1. Yeah, so that is the rate limiting enzyme of fatty acid oxidation. This I've told you in the last session, which is on fatty acid synthesis as well. So what is the rate limiting enzyme of fatty acid oxidation? It is carnitine acyltransferase 1. And this acylcarnitine enters into mitochondria, but as long as it is with the carrier, it can go back to the cytoplasm. So I want you to remove the carrier and attach CoA. That is done by carnitine acyltransferase 2. That forms acyl-CoA within the mitochondria. Now no other go, it gets oxidized. Okay.
Question 3. What could be the possible diagnosis for a patient who exhibited tendon xanthomas, with his lipid profile indicating serum cholesterol levels of 283 mg per deciliter and serum LDL levels of 202 mg per deciliter, and subsequently initiated on lovastatin treatment? Your options are. And the correct answer is familial hypercholesterolemia. Here's the explanation.
Now tell me, which hyperlipoproteinemia presents with an isolated elevation of cholesterol? Logically, what will you call it as? There is only an elevation of cholesterol, right? So what will you call it as? You will call it as familial hypercholesterolemia, which is type 2A. So in which hyperlipoproteinemia do you see only an elevation of cholesterol? That is type 2A. And that is why it is called as familial hypercholesterolemia. And this familial hypercholesterolemia is caused by the defect of LDL receptor. Don't you think it's logical? When LDL receptor is defective on the tissues, if LDL receptor is defective on the tissues, what will not be cleared off from the circulation? LDL will not be cleared off from the circulation. So LDL stays back in the circulation and LDL carries cholesterol. So that will present as familial hypercholesterolemia. And this is an autosomal dominant condition with gene dose effect. What do you mean by autosomal dominant condition with gene dose effect? When will you call a condition as autosomal dominant condition? When even one allele is defective, if you see the presentation, that is when you call it as autosomal dominant condition. So why am I calling hypercholesterolemia, familial hypercholesterolemia as an autosomal dominant condition with gene dose effect? It means when one allele is defective, the condition will be relatively milder. Whereas when both the alleles are defective, the condition will be severe. That is why it is called as autosomal dominant condition with gene dose effect. And in familial hypercholesterolemia, when one allele of LDL receptor is defective, you call that person as LDL receptor defective individual. In which case, LDL receptor activity will be less than 25% of normal. Whereas when both the alleles of LDL receptors are defective, then you call that individual as LDL receptor negative person. Do you understand the difference? One person is just a defective person, the other person is a negative person. In which case, the LDL receptor activity is less than 8% of normal. That is why it's called as autosomal dominant condition with gene dose effect. And that is type 2A, familial hypercholesterolemia.
Question 4. A young girl presented to the OPD for examination. She is tall and thin with slender fingers and dislocated lenses. Workup revealed cystathionine beta synthase deficiency. Which amino acid supplement is required in this case? Your options are. And the correct answer is cysteine. Here's the explanation.
Homocysteinuria causes accelerated atherosclerosis. It causes accelerated thrombosis. And they present with skeletal deformities. Skeletal deformities like flat foot, Charlie Chaplin gait, ectopia lentis. So why are you worried about homocysteinuria? Homocysteinuria is present with accelerated atherosclerosis, accelerated thrombosis, skeletal deformities like flat foot, Charlie Chaplin gait, and ectopia lentis. So if you see a patient with a young patient presenting with stroke, a young patient presenting with myocardial infarction, please estimate homocysteine levels in that person. Now, to understand causes of homocysteinurias, you will have to know about normal homocysteine metabolism. Because homocysteine is a sulfur-containing amino acid, this homocysteine can have two fates. It can get converted to one or the other sulfur-containing amino acids. What are the other sulfur-containing amino acids? Cysteine and methionine. So homocysteine either can become cysteine or it can become methionine. What is the major fate of homocysteine? Major fate is that it gets converted to cysteine.
Question 5. In the diagram provided, which enzymes are identified as E1 and E2, responsible for the conversion of pyruvate to acetyl-CoA? Your options are. And the correct answer is pyruvate dehydrogenase and dihydrolipoyl transacetylase. Here's the explanation.
Now, the most important fact about the subunits of PKH complex. This fact has been asked as a one-liner. This fact can be asked as an image-based question. In the latest NEET PG, there was an image where they showed you 3 circles asking you what is E1, what is E2, what is E3. So, what are the 3 subunits of PKH complex? E1, E2, E3. E1 is named after the whole enzyme complex. It is pyruvate dehydrogenase. Though it is called as pyruvate dehydrogenase, it decarboxylates pyruvate. So it's also called as pyruvate decarboxylase. E2 is dihydrolipoyl transacetylase. E3 is dihydrolipoyl dehydrogenase.
Question 6. Which of the following is true regarding competitive inhibition? Your options are. And the correct answer is increasing substrate concentration can overcome competitive inhibition. Here's the explanation.
Usually when there is an enzyme and a substrate, this enzyme and substrate react together to form enzyme-substrate complex. And this enzyme substrate complex breaks to form either enzyme and substrate, or it forms enzyme and product. Do you understand this? When enzyme reacts with the substrate, it forms enzyme substrate complex. This enzyme substrate complex either breaks to form enzyme and substrate back, or it forms enzyme and product. This is a normal enzyme catalyzed chemical reaction. Now, what happens in the presence of a competitive inhibitor? A competitive inhibitor is a substrate analog. It is exactly like a substrate. So, it competes with the substrate to go and bind to the enzyme's active site. Do you understand this? What is a competitive inhibitor? It is a substrate analog. It competes with the substrate to go and bind to the active site of an enzyme, and it forms enzyme inhibitor complex. And this does not allow the product to be formed. And as I told you previously, in the case of competitive inhibition, it is just a difference in the probability. When there is a competitive inhibitor, it reduces the probability that the substrate goes and binds to the enzyme. It reduces the probability that the enzyme substrate complex is formed. It reduces the probability that the product is formed. If it is just a reduction in the probability, tell me what will happen when you increase the substrate concentration. When you increase the substrate concentration, the substrate will win the competition. The substrate will go and bind to the enzyme and the product will be formed. That is why I told you, in the presence of a competitive inhibitor, increasing the substrate concentration will overcome the inhibition. And if you manage to overcome the inhibition, what is happening to Vmax? Vmax is unaltered. Somehow you have overcome the inhibition, so Vmax will remain unaltered. But to reach that Vmax, you have to increase the substrate concentration. So tell me what will happen to Km. Km is a substrate concentration half maximal velocity. Here you have provided more substrate. So Km is increased. So in the presence of a competitive inhibitor, Vmax remains normal but Km is increased. And this will be evident in a Lineweaver-Burke plot. So all of you tell me, how do you draw a Lineweaver-Burke plot? I am going to draw it again. And I want you to repeat it along with me. How is this Lineweaver-Burke plot drawn? What is drawn along y-axis? Along y-axis it is 1 by V. Along x-axis it is 1 by S. And what will you get? You will get a straight line. And if you extrapolate the straight line cuts the y-axis at 1 by Vmax and it cuts the x-axis at minus 1 by Km. And this is the Lineweaver-Burke plot of the enzyme and the substrate in the absence of any inhibitor. Now, what happens in the presence of a competitive inhibitor? In the presence of a competitive inhibitor, Vmax remains unaltered. I am going to show you the effect of an inhibitor as a red line. So, the red line, Vmax remains unaltered. But what happens to Km? Km is increased. When Km is increased, 1 by Km will be decreased. So 1 by Km is decreased, it is close to 0. So if you see a line like this, in the presence of an inhibitor, if you see a line like this, which means there is normal Vmax but Km is increased, then that tells you that that is a competitive inhibitor. Got it? So have a look at this. This is how the graph will be in the presence of a competitive inhibitor. The last line is in the absence of any inhibitor. Can you see that being marked? No inhibitor. This is the line. And in the presence of some unknown inhibitor, if you have this as the line, do you see that in this case, Vmax is the same? But what is happening to Km? 1 by Km is reduced or Km is increased. So what kind of an inhibition is this? This is a competitive inhibition.
Question 7. Which of the following is true about the structural organization of proteins? Your options are. And the correct answer is tertiary structure is three-dimensional. Here's the explanation.
It's very simple. Tertiary structure of a protein is defined by how a polypeptide chain gets organized in three-dimensional space to form functional domains. Because all this while, all that you have done is you took primary structure, which is just amino acid sequence. And in this primary structure, you introduced ordered units at various places. That is called a secondary structure. So what you have till now is This is a long polypeptide chain with ordered units at various places. Till now, they don't have any functional domains. So what are you going to do next? You take this secondary structure with ordered units and you fold it in a three-dimensional space. That is when they tend to have functional domains, and that is called as tertiary structure.
Question 8. What is the probable reason for the worsening of neurologic symptoms in a patient who initially showed improvement in blood picture after receiving a dose of 5 mg folate for peripheral neuropathy and macrocytic anemia? Your options are, and the correct answer is folate therapy using the B12 stores. Here's the explanation.
Methylfolate trap is in B12 deficiency. Yeah, in B12 deficiency, there is functional deficiency of tetrahydrofolate as well, because all tetrahydrofolate will be trapped as methyl tetrahydrofolate because of low activity of methionine synthase. Yeah, because of low activity of methionine synthase. If you know all these facts, it's more than enough as far as methylfolate trap is concerned. So I'm going to repeat this. What is methylfolate trap? In B12 deficiency, all tetrahydrofolate will be trapped as methyl tetrahydrofolate. That causes functional deficiency of tetrahydrofolate because of low activity of methionine synthase. So now do you understand why B12 deficiency presents with macrocytic anemia?
Question 9. Which of the following deficiencies leads to an elevation of glutamine levels in the bloodstream? Your options are, and the correct answer is ornithine transcarbamylase. Here's the explanation.
Ornithine transcarbamylase is defective. That is called as type 2 hyperammonemia. And this type 2 hyperammonemia is X-linked recessively inherited. This you have to remember, like Fabry's disease. Yeah, like Fabry's disease or like G6PD deficiency. Even type 2 hyperammonemia is X-linked recessively inherited. And in this condition, initially what accumulates? Just have a look at this flow chart and tell me. When ornithine transcarbamylase is defective, first carbamyl phosphate accumulates. This carbamyl phosphate in a feedback mechanism inhibits CPS1. So ammonia accumulates. This is when what will you observe? High ammonia, high glutamate and high glutamine.
Question 10. True regarding mitochondrial genome. Your options are, and the correct answer is maternal inheritance. Here's the explanation.
Mitochondrial DNA. The first property of mitochondrial DNA is that it is a closed circular double-stranded DNA. Similar to plasmids of prokaryotes, your mitochondrial DNA is a closed circular double-stranded DNA. So we actually consider a mitochondria as a prokaryote which has gained entry into eukaryote. And because we later found out that this prokaryote provides us with some survival advantages, we have allowed it to get into symbiosis. So it is a closed circular double-stranded DNA. Point number 2. Though mitochondria has got its own DNA, not all proteins of mitochondria are coded by the same. Mitochondrial DNA codes only for 13 proteins of electron transport chain. It codes only for 22 tRNAs. It codes only for 2 ribosomal RNAs. Put together, how many genes do you find in mitochondrial DNA? Totally, we have only 37 genes, which means your mitochondria is not self-sufficient. Just because mitochondria has got its own DNA, it does not mean all proteins of mitochondria are coded by the same. Most of it are again coded by the nuclear DNA. Point number 3 is the genetic code in mitochondrial DNA is quite different. Because what is usually UGA? Have you learnt about UGA? Yeah, we have learned that UGA is a stop codon. But the same in mitochondria codes for tryptophan. So UGA is not a stop codon, but it codes for tryptophan. Usually AGA and AGG code for arginine. But both these act as stop codons in mitochondrial DNA. So AGA and AGG do not code for arginine, but they act as stop codons in mitochondrial DNA. That is why we say our genetic code is universal, but with few exceptions. Those exceptions are related to mitochondrial DNA. Point number 4 is mitochondrial DNA is more prone to get mutated. Reason is DNA polymerase gamma, which synthesizes mitochondrial DNA, does not have proofreading and repair mechanism. I will ask you this in another 10 to 15 minutes when we start discussing about the various types of DNA polymerases. If I ask you which DNA polymerase synthesizes mitochondrial DNA, what should be your answer? It is DNA polymerase gamma, which does not have proofreading and repair mechanism. Okay, and such mutations are only maternally inherited. Why do you think mitochondrial DNA mutations are only maternally inherited? It is because during the zygote formation, all that the sperm contributes with is its pronucleus. Don't we know that? All that the sperm contributes with is its pronucleus. The remaining part of the sperm will be shed down. And only here you have mitochondria. So sperm mitochondrial DNA does not even take part in zygote formation. So if that sperm mitochondrial DNA has got any mutations, will it be inherited by the zygote? No. So mitochondrial DNA mutations are only maternally inherited.
Don't forget to like and subscribe if you found the video interesting. See you in the next one.